Hello everyone. Welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. Okay, so without wasting any time, let's get started. Coming to the definition, cell cycle progression is the ordered, regulated sequence of events by which a cell grows, duplicates its DNA, and divides to produce two genetically identical daughter cells. A pretty straightforward definition, and it is essential for growth, development, tissue repair, and maintenance of cell populations. Now, just look at this diagram. Once immediately when you view this diagram, you may find it complicated or you may not, but now we are going to discuss each phases, and once after discussing that phases, if you revisit to this diagram, it will be very simple for you, and just draw this diagram in the exam. It will be uh, high yielding for you because this is from Robbins. Okay, so let's discuss one by one. Coming to the first phase, that is G1 phase. See, there are basically interphases and M phases in a cell cycle. Interphases includes G1, S, and G2, and the M phase is separate one. G stands for gap, so gap phase, first phase, second phase, like that, and S is for synthesis, and M is for mitosis. So G1 phase is nothing but first gap phase. So this is period of cell growth, and there is increase in the cell mass. So basically here synthesis of RNA, proteins, enzymes, and organelles takes place, and centrosome duplications also begins in this particular phase. And here the cell assesses the availability of nutrients, growth factor stimulation, cell size, and DNA integrity. So it contains restriction point. What is restriction point? Basically, because see, what happens is, in order to cell to commit division, before, like, for initiating, there are requirement of growth signal. Once it crosses the restriction point, it does not receive any more growth signal, and it is committed to division. So also during this phase, the cells can also exit to the G0 phase, which you basically call as quiescent phase. You would have you would have listened about this during your PU phases, or 11th or 12th, the quiescent phase, basically the resting phase. So some cells can re-enter from G0 to G1, and some cells may not. So there are three types of cells, like labile cells, stable cells, and permanent cells. Labile cells are those which undergo continuous division. Okay, for example, skin cells, so they re-enter easily. Whereas stable cells are those cells which will re-enter only if there is some necessity, if there is some trauma, like hepatocytes. If liver undergoes some any damage, it can regenerate. So those are stable cells. And there are some permanent cells which cannot re-enter, means they do not divide, so the healing occurs only by fibrosis. So even a minor damage also results in fibrosis. So examples are neurons. So this is about G1 phase. Now coming to the next phase, that is S phase. So S phase, here DNA replication occurs, and each chromosomes form two sister chromatids. Histone synthesis also takes place, and centrosome duplication completes during this phase. And here DNA content doubles, but chromosome number remains same. It's a classic line which is given in NCERT if you remember your NEET preparation phase. Okay, so next it is G2 phase. So here the preparation for mitosis happens, synthesis of microtubules, mitotic spindle proteins, all these things happens, and DNA damage repair also takes place during this phase, and cell checks for DNA damage and incomplete DNA replication. So there are few checkpoints in order to do all these things, and we have covered the checkpoints separately in this video in the upcoming part. Okay, so next M phase. It is the stage of mitosis. So you would have studied all these phases like prophase, metaphase, anaphase, telophase. So just mention it. Prophase, chromosome condensation and spindle formation. Metaphase, it is chromosome align of equatorial plate, and anaphase is sister chromatid separate, and telophase, nuclear membrane reforms. So it is followed by the cytokinesis, which is the physical division of the cell. Next, cell cycle checkpoints. Now this is what I was discussing before. So checkpoints are basically like, see, if the G1 phase, there is everything preparation going on for the replication. Now imagine there some damages occur there, like for example, you go out and the ultraviolet rays falls on our skin cells. Now they are actively dividing labile cells. So because of the ionizing radiation, there may be some damage. Now, if there is some damage, if there is some mutation, if we don't check that, if during the cell division process, if it is not checked, then it may replicate, the mutation will become large, more cells will get the same mutation and the problem may arise. So there is a necessity of checking in between. So there are two major checkpoints. One is G1S checkpoint, which happens between G1 phase and S phase, and one more is G2M checkpoint, which happens between G2 and M phase. If you ask me in these two which is more prominent, it is G1S, because here the division before DNA replication only this is happening. If you consider G2M, there already the replication would have happened, and there is something reading and all those which you would have studied in biochemistry. So if you compare both, this G1S checkpoint is more prominent, but both have their own significance. So coming to the G1S checkpoint, it basically checks the DNA damage, cell size and growth factor signals. Okay, and it activates the P53, which would you would have listened the Guardian genome something. So and P53 is separate big topic. You need a separate video for it. Here I have just explained the basic of it. So P53 activation it does, and P53 basically activates P21 something, and that that does the CDK inhibition. Now, what is CDK inhibition? It is cyclin dependent kinase inhibition. We are going to discuss what is cyclin dependent kinase in the next part of the video. So now it prevents the damaged DNA from replicating. Obviously, whatever the damage have been happened, it have been prevented by the G1S checkpoint. Next, G2M checkpoint. Here it ensures complete DNA replication and absence of DNA damage, and it prevents entry into mitosis if defects exist. Okay, like G2 phase and M phase. M phase is the phase where the cell divides. Okay, so just before the cell is dividing, it just checks whether DNA replication have been completed. Is there any damage? If there is any damage, it does not allow that cell to divide. So that is the major role of these two checkpoints. You have to mention this. These are very important. Next, third one you can mention or you may leave it, that is spindle assembly checkpoint. So it ensures the proper attachment of chromosomes to spindle fibers, and it prevents chromosomal missegregation. Now coming to the molecular regulation of cell cycle proliferation. First thing, cyclins and cyclin dependent kinases. We were discussing there. You may not have understood what it is, so let's see that. So cyclins are the regulatory proteins. Okay, they are the proteins which regulate the cell cycle, whereas cyclin dependent kinases, they are the catalytic enzymes. What happens actually, cyclins are the proteins which are in the inactive form. In order to make them activated, we need some agents which can phosphorylate it. Now you would have studied in the biochemistry the class of enzyme which are involved in the phosphorylation and dephosphorylation are the kinases. So there are some group of enzymes which are called as cyclin dependent kinases which phosphorylate and activate the cyclin. So cyclin CDK complexes drive the cell cycle forward. Next, just make a box. So here there you can see the diagram. There is G1 phase, S phase, M phase, and G2 phase and M phase. So from G1 phase to S phase, and you can also see the checkpoints there, G1S and G2M. So now if it wants to get converted from G1 to S, there are two cyclins which play a major role, that is cyclin D and cyclin E. So the cyclin dependent kinases which activates those cyclins are CDK4, CDK6 and CDK2. So likewise all other cyclins and the respective kinases which activates them are mentioned in the boxes. You have to memorize it. But the important point here is, look there, if it wants to get converted to S phase, we need those cyclins. If there is any defect in it, it may result in something abnormal, something pathogenic. So that thing we have mentioned in the clinical importance or clinical significance in this video, which will come at the end part. So that thing you have to mention. Next, role of retinoblastoma protein. Now, what is retinoblastoma protein? It is a tumor suppressor. So in the early phase of G1, this retinoblastoma binds to E2F. Now E2F is a transcription factor which is present in the cyclin E. you would have seen in the box or the diagram that if a cell has to convert it to G1 phase to S phase, it needs a cyclin called as cyclin E. So, and it needs cyclin D and cyclin E both. So first, what this retinoblastoma protein does is it goes and binds to the transcription factor which is present in cyclin E. Now, as soon as the CDK4 and 6 activates the or phosphorylates the cyclin D, this retinoblastoma gets phosphorylated. No, so this phosphorylated retinoblastoma that releases the transcription factor for which it was bounded. So now what happens is cell progresses from G1 to S phase because the cyclin which was required for the progression of cell from G1 to S phase was cyclin E, which was bounded by the retinoblastoma. As soon as it gets phosphorylated by the cyclin D, it releases the cyclin E, and that cyclin E goes and converts the cell from G1 phase to S phase. So that is the role of retinoblastoma. And last mention the CDK inhibitors. Now CDK inhibitors are two types. One is INK4 family and one more is CIP or KIP family. This name might look little bit complex. You can simply write it as specific and non-specific. Now specific are those, if you remember, while discussing the checkpoints, we said G1S checkpoint is very, very important. So the inhibitors which are going to inhibit the cyclins or cyclin dependent kinases which are involved in the G1S checkpoint are specific. They're going to specifically inhibit those cyclins itself, whereas the non-specific are those which can randomly inhibit any CDKs or cyclins. So that is the difference. So under specific, P15, P16, P18 and P19 comes. Now again, what is P? They haven't given any specific names for these. P is nothing but protein of that number basically indicates the weight of that protein. So P15 means protein of 15 kilodalton. P16 is protein of 16 kilodalton. P53 is protein of 53 kilodalton. It's that simple. So P15, P16, P18, P19 comes under specific, and it inhibits the cyclin D that we discussed, that is specific cyclins, and it act mainly at the G1 phase, whereas non-specific, it broadly inhibit multiple CDKs and it mediates cell cycle arrest after DNA damage. Then role of P53, just mention it because P53 itself is a very big topic. So P53 is basically activated by DNA damage or hypoxia or cellular stress. So what does P53 do? It causes a cell cycle arrest. It temporarily arrests the cell cycle with the help of activation of P21, and it tries to repair the DNA. You would have studied in biochemistry the various DNA repair mechanism. So in the checkpoint, the P53 recognizes if there is some DNA damage, it arrests the cell cycle with the help of P21, and it tries to repair the DNA. If the repair is successful, the cell cycle continues, and if the repair is not possible, there is irreversible damage, then it induces apoptosis of the cell. So that is the gist of role of P53. Then last but not the least, the clinical significance. So if there is any defects in cyclins or CDKs or CDK inhibitors or retinoblastoma or P53, it will lead to the loss of cell cycle control, which causes uncontrolled proliferation, which may lead to cancer. And many anti-cancer drugs act by arresting specific cell cycle phases. So that's all for this video, and I hope this was helpful. So please show your support by liking and commenting, and keep studying. Do well. All the best. Thank you.